We have a really spectacular piece of translational science to discuss today. I mean, this isn't just theory.
Not at all.
This is advanced computation really hitting the wet lab hard and solving a major medical puzzle, one that stumped researchers for, what, a quarter century?
That's right, 25 years. We're looking at a landmark piece of work on Crohn's disease involving the notoriously tricky NOD2 gene.
And the role of applied AI in cracking it. This comes from a team at UC San Diego.
Yeah, and what's really critical about this specific research, I think, is how it moves past the usual hype you hear about AI and drug discovery.
Right, it's not just prediction.
Exactly. A lot of studies use machine learning to, predict outcomes or classify things. This work used it to define a very precise, a targetable biological mechanism.
Okay.
It successfully connects massive transcriptomics data, that's gene expression data, with actual real-world cell biology. It radically shortens the path from just piles of data to a viable therapeutic strategy.
So it's like a blueprint.
It really is. It gives us a potential new blueprint for precision medicine, especially in inflammatory diseases.
Okay, so our goal here today is pretty straightforward. We want to make sure you walk away understanding exactly how this tiny protein, GIV, ended up being the key, how it governs this huge immune response that goes so wrong in Crohn's disease. We're going to unpack the specific molecular mechanism they uncovered.
And see what it means for future research.
Yeah, the new blueprint this might create across different fields.
Okay, so let's start with why this took 25 years. Let's set the stage with the unsolved puzzle of NOD2.
Right, because if you look at Crohn's disease overall, it's fundamentally a problem of balance, isn't it?
It is.
In the lining of the intestine, you've got this battle that's just gone completely haywire. And the main players are these immune cells, macrophages.
Key cells, yeah.
They basically have two jobs. First, they're the fighters. They rush towards any microbial threat. They sound the alarm, trigger these aggressive immune responses.
On the second job.
The second job is being the healers, cleaning up all the debris after the fight, patching up the tissue. But in Crohn's, it seems like the fighters just never stand down.
And the healers can't finish their work. It's stuck in alarm mode.
Exactly.
And for decades, we've known the NOD2 gene is linked to this. It's been firmly established. People with certain variants, certain mutations in NOD2 are much more likely to get Crohn's.
So scientists knew it was involved in disrupting that macrophage balance.
Right, pushing the cells towards chronic inflammation. But the problem was, they didn't know the exact molecular function that was broken. It was just fuzzy.
And if it's fuzzy, you can't target it properly.
Precisely. If you don't know the how, you can't design a specific drug to fix that specific mechanism. You end up stuck with broad immunosuppressants, which have their own issues.
Okay, so the mechanism was elusive. How did the computational team even begin to cut through that fog, to narrow down the possibilities from, I imagine, thousands of genes?
Well, they started by embracing the complexity, actually, what they called the messy biology. Instead of trying to average things out or smooth over the fact that tissues contain mixed cell populations, they used machine learning specifically to define stable cellular states.
So looking for consistent patterns.
Exactly. They analyzed thousands of gene expression profiles from both inflamed and healthy human colon tissue samples. They were looking for patterns, signatures that were highly predictive of that healer state in macrophages.
And that computational work, it didn't just give them a vague idea, did it? It pointed to something specific.
Very specific. It defined a precise 53 gene signature. Think of it as a molecular barcode that marks that crucial macrophage healer population.
53 genes. That's remarkable focus from such complex data.
It really is. And then the critical step. Inside that 53 gene signature, one protein, a scaffolding protein, consistently emerged as a known interaction partner for the NOD2 receptor.
And that was GIV.
That was GIV, also known as Girdin. The AI essentially acted like a sophisticated filter, removing all the biological noise to pinpoint GIV.
I really like the way they described the method. It sounds like a two-step filtering approach.
It was.
First, they demanded stability, right? The gene signature had to reliably separate the healthy from the inflamed tissue across different data sets.
Yep. Consistency was key.
And then the cross-referencing step, checking which of those stable genes were already known from other studies to physically interact with NOD family receptors, like NOD2.
And that cross-referencing is absolutely crucial. The machine learning might give you a list, maybe still hundreds or thousands of possibilities, initially based on expression alone.
Right.
But overlaying that with known biological interaction networks, that's what zeroed in on GIV. It was the only one that consistently fit both criteria.
So the computation acted like a high-powered compass,
Yeah,
basically.
a very precise compass, yeah. Pointing the experimental lab team toward the single most promising, most tractable thing to test.
Okay, so the AI points directly at GIV. What happened when they took that into the lab? What did the physical validation actually show about GIV's job?
So now we can finally get into the mechanism with real clarity. The way to think about it maybe is dynamically. NOD2 is like the cell's security sensor. It's constantly checking for bacterial bits, for threats.
Okay.
And GIV acts like the hand on the brake. It makes sure that once the threat is being dealt with, the inflammatory alarm gets turned off at the right time. It controls the response.
And the research details that healthy partnership quite clearly. In healthy macrophages, there's a specific bit on the NOD2 receptor
A specific region, yeah,
that binds tightly to a part of GIV. It's C-terminal pale, they call it.
it's a one.
And when that partnership is working, when they're bound together, two key things happen efficiently. The cell clears out bacteria properly, and GIV actively helps dial down the main inflammatory switch, NF-kappa B, when it's supposed to.
And NF-kappa B, we should stress, is absolutely central to inflammation. Pretty much all inflammatory pathways run through it.
So when you say it runs hot,
When we say NF-kappa B signaling runs hot, it means the cell is stuck with the red alert button jammed on permanently, which is, catastrophic in the long run.
and here's where the specific Crohn's link comes in so tragically. The most common high-risk variant of NOD2, the one strongly associated with the disease, it's not just a subtle change.
No, it's structural.
It's a mutation that physically chops off that crucial docking region on the receptor, the very part GAV needs to grab onto.
Exactly right. The necessary binding site is just gone. So GIV cannot bind. The hand slips off the brake, essentially.
And NF-kappa B signaling runs wild.
Completely unchecked. That's the first major failure. Excessive runaway inflammation because the off switch is broken.
And there's a second failure too, isn't there? It connects back to the macrophage's job as a cleaner upper.
Yes, this is critical. The broken NOD2 GIV interaction doesn't just leave the alarm blaring, it also slows down the cell's ability to actually kill and dispose of the invaders it's engulfed.
And the mechanism for that slowdown is fascinating. It affects a process called phagosome-lysosome fusion.
Right. Think of it like the cell's internal trash collection and incineration system.
Okay.
The macrophage engulfs the bacteria. That packages the phagosome. But to destroy the contents, it needs to fuse that package with another compartment called the lysosome, which contains all the digestive enzymes. It's the incinerator.
And if NOD2 and GV aren't interacting properly?
That fusion step stalls. It becomes inefficient. It's a physical kinetic bottleneck. The trash bags pile up because they can't get to the incinerator quickly enough.
Wow. So you end up with a cell that's aggressively shouting fire with inflammatory signals, while at the same time it's failing to efficiently put out the fire or clear the debris.
That's it, exactly. That combination, excessive signaling plus stalled cleanup, perfectly captures the cellular chaos of the chronic inflammation you see in Crohn's disease.
And that is really precise. You're translating a specific genetic mutation, that NOD2 truncation, into a clear, measurable dysfunction at the cellular level.
And once you have a mechanism that clear, that elegant, you absolutely have to test it rigorously. You can't just rely on the computer model.
Which they didn't?
No, they moved straight to the biological validation, which honestly is often the most compelling part of this kind of computational biology work.
So once GIV was flagged by the AI, did the lab results immediately click, or was there some initial skepticism? What was the first key experiment they did?
The first key experiments really confirmed the paradox beautifully. They took standard cultured macrophages, immune cells grown in a dish, and used genetic tools to completely remove GIV. So GIV-deficient cells.
Okay, what happened?
Well, just as the broken brake idea predicted, these cells fired off inflammatory signals way too aggressively when stimulated. But, and here's the paradox, they were also much worse at actually clearing bacteria they were fed.
Ah, so it immediately showed both sides of the coin.
Exactly. It was immediate validation that GIV was essential for that proper, balanced function controlling the alarm and enabling efficient cleanup.
The source material also mentions they used electron microscopy. That's getting right down to visually confirming the mechanism, isn't it?
Absolutely. You can't get much more direct than looking at the structures themselves. The microscopy provided the visual proof of that stalled cleanup.
What did they see?
They literally saw fewer fusion events between the phagosomes holding the bacteria and the lysosomes, the incinerators, in those GIV-deficient cells.
So visual confirmation.
Visual confirmation that the cell can grab the debris okay, but the process of actually destroying it is bottlenecked. The incinerator door isn't opening often enough or fast enough. That physical inefficiency explains the stalled healing process.
Okay, cell culture confirmed it. Microscopy confirmed the mechanism. The final step was showing it mattered in a whole organism, right? Using mouse models.
That's the crucial in vivo step, yes. They used mice and induced a form of colitis, gut inflammation, chemically. Now, we always have to be careful. Mouse models aren't perfect replicas of human Crohn's.
Sure, standard caveat.
But for testing a specific mechanism like this, they're invaluable.
And what did those models show?
The results were very clear regarding the mechanism. Mice, whose myeloid cells the family macrophages belong to, were engineered to lack GIV, they developed significantly worse colitis. The inflammation was more severe without GIV.
That supports the idea that GIV is protective?
It does.
Oh.
But the real clincher, the ultimate proof for the mechanism, came when they tested a known treatment ligand, something called miramyl dipeptide, or MDP.
What does MDP normally do?
MDP activates NOD2, and in normal mice, giving them MDP actually protects them somewhat against this chemically induced colitis. It boosts the right kind of response.
Okay.
But in the mice lacking GIV, that protective effect of MDP vanished completely. It did nothing.
Wow, that's a perfect control experiment.
It really is.
It proves the entire protective pathway initiated by activating NOD2 depends entirely on GV being there to interact with it. If GV's gone, activating NOD2 doesn't help.
Exactly. So if you're tracking the flow of evidence, this wasn't just, one lucky find. It was this really rigorous step-by-step process.
Yeah, it sounds like it.
From the computational screen, using transcriptomics to define that target profile.
Right.
To the network mapping overlay that prioritized GIV, then through the cell biology and the microscopy that explained the kinetic bottleneck.
The stalled cleanup.
Yes. And then finally, the mouse models tying that specific mechanism directly to the disease severity and the response to treatment.
That really is the ideal roadmap for this kind of research.
It is. It shows how you can take a discovery initially made in silico, on the computer, and turn it into a solid biological fact that's genuinely ready for drug development efforts.
So let's talk about that. What does this discovery actually mean for patients? People living with Crohn's, often dealing with those broad immunosuppressants. How does this shift the conversation?
Oh, it shifts it dramatically. It moves us away from just dampening the entire immune system and towards the idea of mechanism repair.
Fixing the specific broken part.
Exactly. The immediate hope is developing drugs that target the NOD2-GIV interaction itself. Researchers are already actively exploring different angles.
Like what?
Things like small molecule drugs that might physically stabilize that docking interface, help NOD2 and GV stick together properly, even if the NOD2 is mutated, or maybe designing peptides that mimic the crucial part of the GV tail to sort of substitute for its function.
Or biologics, maybe.
Or biologics, yeah, perhaps antibodies or other proteins designed to specifically promote that NOD2-GIV bond. The point is, it's upstream intervention.
Trying to fix the cause.
Trying to fix the cause, not just constantly mopping up the downstream mess of inflammation.
And this feels like true precision medicine finally coming into focus. For patients who carry that specific NOD2 variant, the one that truncates the protein,
they become ideal candidates.
They're the perfect group for clinical trials targeting this specific newly discovered interface.
Absolutely. And furthermore, the research team suggests a potentially powerful tool for those future trials. We might be able to stop relying solely on, subjective symptom reports from patients. They propose using composite scores, derived from biopsies perhaps, that actually measure the healer versus fighter state of the macrophages in the gut lining.
Ah, like an objective readout.
Exactly, an objective pharmacodynamic readout. It means we could measure if a potential drug is actually fixing the underlying cellular mechanism, pushing macrophages back towards the healing state, not just temporarily reducing symptoms.
That could make trials much more informative.
Definitely.
Now, you mentioned earlier this isn't just about Crohn's. The real impact might be broader, as a kind of blueprint.
I really think so. This provides a fantastic model for how AI-driven research can work effectively across medicine. A common critique of AI in biology is that it sometimes just chases abstract benchmarks or finds correlations without explaining why.
Right, finding patterns without necessarily finding causes.
Precisely. But this case treats the machine learning model as that compass we talked about. Its job was to point directly toward a single, tractable, and importantly, verifiable experiment in the lab.
And that framework, that process, seems like it could be adapted elsewhere.
Highly adaptable, I'd say. Whether you're studying chronic inflammation in, say, atherosclerosis, or dysfunction of microglia immune cells in neurodegenerative diseases like Alzheimer's,
Or even cancer.
or the tumor microenvironment in oncology, yeah. You could potentially lift this playbook. It represents the difference between just generating a dashboard full of
Yeah.
interesting correlations.
And actually identifying a druggable target with a known mechanism.
Exactly. The process seems clear now. Define the stable cellular state associated with the disease, use computation informed by biology to find the likely mechanistic switch, the GIV equivalent for that disease,
It is.
and then test that switch rigorously in the lab and in relevant models. Define the healer, find the switch, test the switch.
Which brings us right back to where we started today. That long-debated kind of fuzzy role of the NOD2 gene in Crohn's. It finally has a precise answer.
It does.
It's this partnership with GV. That interaction is the brake pedal that's supposed to keep macrophages in the healing lane. Break that bond, lose that interaction.
And inflammation runs wild while the cleanup crew stalls.
Yeah.
And the fact that the structural footprint of this interaction is now becoming clearer is, really excellent news for drug designers. It gives them something concrete to aim for.
But it's not easy, right? Drugging protein interface.
Oh, it's notoriously difficult.
That's
It's
it.
definitely a challenge. But here's a final thought, perhaps a provocative one for listeners to consider. This breakthrough sits right at the crossroads of immunity and the microbiome.
Which is central to Crohn's disease.
Absolutely. The gut environment, the bacteria there, they're constantly interacting with these immune cells. So the big challenge, and maybe the next great leap, might lie in combination therapy.
Meaning what, specifically?
Well, imagine pairing a drug that stabilizes that NOD2 GIV interface, fixing the macrophage's internal machinery with something else. Maybe a narrow-spectrum antimicrobial that targets specific bad actor bacteria in that individual's gut.
Or maybe a prebiotic.
Or a specific prebiotic or engineered phage therapy designed to reshape the microbial community in a beneficial way. A two-pronged attack.
Acknowledging it's a complex balance problem.
Exactly. It's not just a single cell failure. It's an ecosystem problem. Fixing the host response and addressing the microbial trigger or imbalance simultaneously.
That really is what translating these complex algorithmic findings into real human biology looks like. Moving beyond the single target idea.
It is. So we'd strongly encourage you, the listener, to keep an eye on this field. Track those clinical trials that will hopefully start measuring macrophage healing states and specifically looking for NOD2 GIV function restoration.
Because this feels like a very tangible step towards therapies that target the root cause of the disease, not just managing the symptoms.
A really promising step forward.